Good morning, everyone. It's a privilege to introduce our Grand Rounds lecturer today. Michelle Lebeau is the Cancer Center Director at the University of Chicago Comprehensive Cancer Center. She did her undergraduate work as a Boilermaker at Purdue and her graduate work at University of Illinois Chicago, and really has had a very significant research career in the area of cancer genetics and cytogenetics. She leads the Cancer Cytogenetics Laboratory at University of Chicago, and a lot of the work she's best known for really relates to her title of her talk today. She really has been a leader in looking at molecular abnormalities that are repetitive in human tumors, particularly in the therapy-related myeloid malignancies. Here at Winship, we know Michelle best as a member of our external advisory committee and really has been an invaluable advisor to Winship. Michelle led a special external advisory committee meeting a couple years ago on comprehensiveness. And the C in Winship's Comprehensive Cancer Center, I give Michelle as much credit for as anyone. She's really been an invaluable advisor to us, very thoughtful. And don't mistake Michelle's soft-spoken, quiet manner for a lack of knowledge and clarity and leadership, because she's really known around the country and highly sought after as an advisor to cancer centers. But it's great to have Michelle here. I think she'll be meeting with some of you later in the day, and welcome, Michelle.
Good morning, and thank you, Wally, for such a lovely introduction. It's really a pleasure to be here today. As Wally said, I've come here under a different capacity, and I have been so impressed with what Wally and his colleagues have done in building the Cancer Center. I'm used to seeing Wally up here at this podium speaking, so it's a little bit of a change now to be here visiting you, but I really appreciate the opportunity. And I wanted to talk about a disease, therapy-related myeloid neoplasms, that we have been working on for several decades now at the University of Chicago. And I think most of you recognize that due to the aging population, the increase in cancer patients and improvements in cancer therapy, that the number of cancer survivors has been growing dramatically. And it's estimated that there are now almost 18 million cancer survivors in the US. But one of the consequences of successful cancer therapy is that the incidence of therapy-related and secondary cancers are increasing. And this is, again, because of the increased number of patients who are surviving. The disease that we study is therapy-related myeloid neoplasms. And this is really an unfortunate late effect of cytotoxic therapy in patients who are treated for a primary cancer with chemotherapy and or radiotherapy. We now recognize that there are very distinctive clinical and cytogenetic subsets of therapy-related myeloid neoplasms. The major subset arises in patients who are treated for a primary malignancy with alkylating agents. And these patients have a latency of about 5 to 7 years from treatment of the primary malignancy to the development of the secondary myeloid neoplasm. They often present with the therapy-related myelodysplastic syndrome that rapidly progresses to AML. And the disease is characterized typically by deletions of chromosome 5 and loss of chromosome 7 or del7q. And this is a very aggressive disease, and the median survival in our population of cases is 8 months. There's a second major subset of myeloid neoplasms that arises in patients who are treated with TOPO2 inhibitors. And these patients present with an acute leukemia 2 to 3 years after treatment for the primary malignancy. And it's typically characterized by the recurring translocations involving MLL or KMT2A or RUNX1, similar to the translocations that we see in de novo AML. And this group has a slightly better, but still relatively poor prognosis. So, as I've already said, the significance of studying this disease is that the incidence is increasing, and it's beginning to represent a significant survivorship issue for our cancer patients. But importantly, it also provides clues to leukemogenesis. We don't know all of the causes for cancers, but in this case we know that the disease, incidence of the disease is influenced by the cytotoxic therapy that they receive. It also allows us to study the role of genetic susceptibility to cancer and the role of other environmental influences. So the risk of therapy-related myeloid neoplasms is shown in this slide, and this shows the incidence after chemotherapy. The incidence is about less than 1 per 100,000 individuals, but it accounts for about 15% of new MDS and AML cases, and the median age at diagnosis is 64. And chemotherapy is a significant factor in that it increases the incidence of this disease almost 5-fold. Now, we can see therapy-related myeloid neoplasms in rising for patients who are treated for almost any primary malignant disease. However, the incidence is, the majority of our patients are treated for breast cancer, or non-Hodgkin's lymphoma, or multiple myeloma and Hodgkin's lymphoma, so breast cancer and the heme malignancies. But we're also seeing another group that has emerged recently, patients who are treated with for cervical cancer, ovarian cancer, and prostate cancer. And I think many of you will already realize that the incidence is quite high in patients who are treated for soft tissue sarcomas, with an incidence of about 11%. So our group has been studying therapy-related myeloid neoplasms, as I mentioned, for several decades, and we've focused on defining the clinical and cytogenetic features, some of the molecular networks that are involved. We've been studying the pathobiology of deletions of 5q and developing mouse models for this disease, which I'll talk about today, as well as identifying therapeutic targets and therapeutic approaches, and genetic predisposition. So I'd like to touch on many of these points today. And the major points that I'd like to make are that a subset of patients have germline susceptibility mutations that put them at greater risk for this disease. Therapy-related myeloid neoplasms are genetically similar to other high-risk myeloid neoplasms. I think this is something that those of you who treat patients leukemia patients already recognize, that high-risk de novo AML is very similar to therapy-related myeloid neoplasms. The deletion of 5q results in haploinsufficiency of multiple genes on 5q. This is a contiguous gene syndrome, and cooperating mutations of P53 also participate in the pathogenesis of this disease. Aberrant Winch signaling in both bone marrow niche cells and hematopoietic cells drives leukemogenesis in this disease. And cytotoxic therapy has effects on both the bone marrow niche and the hematopoietic stem cells. We know that it, from elegant studies that I'll present from the work of others, that it promotes clonal selection of pre-existing mutant hematopoietic stem cells, but it also induces leukemogenic mutations. And finally, targeting the Wynt pathway may be an effective therapeutic approach in some cases with this disease. So what are the risk factors for therapy-related myeloid neoplasms? The type of primary malignancy, the type of cytotoxic therapy, and cumulative doses received are risk factors. There have there's a very large body of literature looking at inherited risk factors such as polymorphisms in proteins that are involved in drug metabolism and DNA damage response and repair. This is a little bit cloudy in the sense that there are as many positive papers showing a correlation with variants in these genes as well, and other papers that show no correlation. Several groups, including ours, have recently shown that a subset of these patients have mutations in inherited cancer susceptibility genes, most commonly BRCA1 and 2 genes and P53. The phenomenon of clonal hematopoiesis of indeterminate potential, or CHIP, has received a lot of attention recently and has emerged as a major risk factor for therapy-related myeloid neoplasms. CHIP is a phenomenon where we see somatic mutations arising in hematopoietic stem cells. They're relatively rare in younger individuals, but ultrasensitive sequencing has shown that they are present in up to 95% of individuals who are older than 50. The most common involve genes like DNMT3A, ASXL1, TET2, and P53. And these are the same genes that we see mutated in hematologic malignancies and myeloid neoplasms. So it's estimated that patients who have CHIP have an increased risk of hematologic malignancies. It's been proposed that CHIP may be the first biomarker for predisposition to developing myeloid leukemias. It's estimated that the presence of CHIP has a positive predictive value of 30% and a negative predictive value of about 90%. But I think people are reluctant to really put this into clinical practice because we don't know the exact risk associated with each of the specific mutations or combinations of mutations. So the phenomenon, what's believed to happen is that CHIP may be an early initiating mutation in critical genes, shown in the green, and that these, a clone, a hematopoietic stem cell, that there can be clonal expansion and an increase in the population of cells that have that mutation, and they acquire additional subsequent cooperating mutations and give rise to a malignant disease. So we'll come back to talk about these processes in a short while. Okay, so one of the reasons that I think it's important to understand who is at risk is the concept of risk assessment and prevention. And in the past, we've always thought about risk assessment really as a one-size-fits-all. But I think we're moving to different models now where we can divide patients into different populations based on high risk or low risk. And the importance of this is that there may be approaches that we can use by identifying patients who might be at increased risk, those who have CHIP, and modifying primary therapy using risk-directed screening, using targeted molecular chemoprevention such as niche-based therapy to create reduce the incidence of CHIP and to create a healthier hematopoietic stem cell population. So coming back to the concept of cancer predisposition, years ago we recognized in our series of patients with therapy-related myeloid diseases that the myeloid leukemia was not the second cancer for many of our patients, and that about 10% of our patients had had 2 or more cancers prior to developing therapy-related myeloid neoplasms. So of course that immediately suggested to us and others that these patients may have familial susceptibility mutations. And until recently, it really wasn't possible to test this hypothesis. But my colleague, Jane Chirpik, using a series of patients, identified 109 patients in our series of therapy-related cases that had myeloid neoplasms following breast or ovarian cancer. And 53 of those patients had germline DNA available for sequencing. And she performed that analysis using a panel of ovarian and breast cancer susceptibility genes and found that 23% of these patients had a deleterious mutation in BRCA1 or 2, P53, PALB2, and CHEK2. So these are patients that are important to identify in advance. And we've also looked at other patients who have myeloid neoplasms either developing at a younger age or following sarcomas, and again see a very high incidence of familial cancer susceptibility, germline mutations in these patients. So this is a population that we may want in the future to identify in advance. So turning now to the genetic abnormalities that we see in myeloid neoplasms, this slide illustrates the frequency of cytogenetic abnormalities that are used for risk-adapted therapy in AML, based on our series at the University of Chicago. And the only point I'd like to make is that de novo AMLs tend to have a high frequency of the recurring translocations that we recognize in this disease, whereas they have a lower frequency of abnormalities leading to loss of chromosome 5, 7, or both. The reverse is true of therapy-related myeloid neoplasms. 70 to 75% of these cases have abnormalities leading to loss of chromosomes 5, 7, or both. And these are the high-risk aggressive diseases. And a very small percentage of patients have the recurring translocations. And this is the subgroup that are treated with topo-2 inhibitors, whereas this group of patients are the group arising after alkylating agent therapy primarily. So looking at the distribution of cytogenetic abnormalities in therapy-related myeloid neoplasms is shown here. And gain of individual chromosomes is shown in green and losses in red. And the only thing I wanted you to appreciate in this case is that there's a lot more red than there is green. So this is a disease characterized primarily by the loss of genetic material. So when we started this analysis, we already knew about some of the common abnormalities, deletions of chromosome 5, loss of chromosome 7, trisomy 8, and loss of 17p that characterize this disease. But by studying a larger number of patients, we were also able to recognize the a non-random pattern of some additional abnormalities, loss of 12p, loss of 13, loss of 16, 18, and 20q. And it turns out that this is a very distinct pattern that is associated with abnormalities of chromosome 5. And if we look at the continued genomic analysis of this disease using SNP arrays, we see that this is a complex disease with about 7 copy number changes per case. But it turns out that we always think about these diseases as being genetically unstable and complex, but it's because of abnormalities of chromosome 5. Patients who have abnormalities of chromosome 5 have on average 14 copy number changes and complex karyotypes, whereas patients who have loss of chromosome 7 have only 2.5. Patients who have abnormalities of chromosome 5, about 80% of them have P53 loss or mutations. So that may account for the genetic instability in this disease. We've gone ahead and done whole exome sequencing on a number of these cases in collaboration with Jim Downing. And again, we find that patients with deletions of chromosome 5 have mutant P53, but relatively few mutations overall. Last year, 2 years ago, there was a very elegant study published by Lindsley and Ben Ebert. And I think they defined very nicely three subtypes of therapy-related myeloid neoplasms. The first subtype is similar to AML that arises after MDS. And these patients tend to have high frequency of mutations in the spliceosome genes. The second subtype looks like de novo AML, and they tend to have mutations in NPM and the recurring translocations. And then the third subtype is the P53 mutated subtype that I've just mentioned to you. They have a high frequency of deletions of 5 and complex karyotypes, and very few other mutations. So originally, people concluded that cytotoxic therapy is not causing these mutations because of the low frequency that are seen in this subtype, but also because we don't see a high frequency of transition mutations, a type that would be induced by alkylating agents. But I think that's a little bit misleading because we're not assessing the role that the alkylating agents may cause in causing double-strand breaks that lead to the acquisition of these complex cytogenetic abnormalities. So I'd like to tell you what we've done to study the deletions of chromosome 5. And I'm embarrassed to say that we started this work almost 2.5 decades ago and haven't completely figured it out yet. When we started, there were no genes, there were about 10 genes that had been mapped to the entire chromosome 5. So we had to do a lot of genetic mapping. But some of the work that we did was, we hypothesized that the loss of one or more genes on 5q contributes to the development of myeloid neoplasms. And some of the questions that we've asked over the years are, which gene or genes are important? What are the functional consequences of loss? Do mutations on other chromosomes cooperate? What's the role of cytotoxic therapy, and what is the role of the bone marrow microenvironment? And I'd like to give you a quick kind of summary of some of the things that we've done over the years. So when we first began to map the deletions of chromosome 5, we mapped the deleted segments using fluorescence in situ hybridization. And then we advanced and also applied SNP arrays to study these cases. And what I'd like to point out to you here is that the deletions are interstitial, meaning that there's 2 breaks, and that the central portion of 5q is retained and the distal portion of 5q is typically retained in these cases. An important point that we learned from gene expression studies is that you see that the pattern of deletion corresponds very nicely with a decreased expression of the genes that are encoded on this portion of chromosome 5. So many of the genes on 5q are subject to a gene dosage effect, where you lose 1 copy and you now have a 50% reduction in the level of protein product. So based on the work of ourselves and others, we've learned that it's haploinsufficiency that drives the development of deletion 5q disorders, and that there are a number of genes that are haploinsufficient on this chromosome. By mapping critical deleted segments, we've identified 2 regions of chromosome 5 that are the minimally deleted regions. And the minimally deleted region in therapy-related disease is here on 5q31 and contains a number of candidate genes. EGR1 is the one that I'll talk about today. There's a second disease called the 5q minus syndrome, or MDS with isolated del 5q, and that involves genes, a commonly deleted segment that's a little bit distal on 5q and involves RPS14 and several microRNAs and casein kinase 1A1. There are some additional genes that map outside of these commonly deleted segments or minimally deleted regions that have been implicated in the disease. And APC is one that I'll talk about today. The problem in studying these disorders is that patients have large deletions. This is the most common deletion seen in almost 70% of cases. So patients lose both of these commonly deleted segments as well as all of the genes in between. And this has made it difficult to identify what gene or combination of genes are the critical ones. But just to summarize what we've learned over the years, I've already told you about the 2 deleted regions. We initially began this work thinking that it was a classical Knudsen 2-hit model. However, we were not able to identify homozygous mutations in any of the candidate genes on the remaining chromosome 5. And it turns out that many of the genes are expressed at 50% levels, and this appears to be haploinsufficiency that's leading to the disease. And it's more complicated because it's loss of multiple genes that contribute. And this is what we know as a contiguous gene syndrome, in which the phenotype is induced by a combination of reduced expression of adjacent genes. And one of the ways that we've been able to map the role of some of these genes is to make mouse models. And we've learned that haploinsufficiency for only a single gene recapitulates some of the features of the disease, but not all. And it's only when we create combinations of mice that are haploinsufficient for multiple genes that we begin to recapitulate the whole disease. So for example, loss of RPS14 and APC result in anemia, whereas these microRNAs result in megakaryocytic dysplasia. Several of the genes loss creates expansion of the stem cells, and clonal dominance seems to be regulated by casein kinase 1A1. So I'd like to tell you this story about the work that we've done to create a mouse model for deletions of chromosome 5. And The first gene that we started with is EGR1. And this is a transcriptional regulator that regulates a number of critical genes, including P21 and P53. It's a zinc finger protein that's a member of the WT1 family. It does regulate stem cell quiescence and retention in the bone marrow. And so we made a knockout model, a heterozygous model, and the mice had absolutely no phenotype. But when we treated them, thinking back about the patients, when we treated them with ENU, an alkylating agent, they developed a myeloproliferative disease with ineffective erythropoiesis. And I'd be remiss if I didn't point out that this work started through a collaboration with your dean, Vikas Sukhatme, who cloned this gene and characterized this gene in normal conditions as well as in disease conditions. The second gene on chromosome 5 that we've studied is the APC gene, which you're all familiar with as a negative regulator of the Winch signaling pathway. But APC has multiple roles. It regulates mitosis and cell migration, and more recently it's believed to play a role in chromosome segregation and in DNA methylation. So we created a conditional mouse model using MX1 Cre so that we could delete one allele in hematopoietic cells. And the mice developed a myelodysplastic syndrome and a severe macrocytic anemia. The third gene is P53, which I think is familiar to all of you. Loss of P53 is seen in about 80% of patients with a del5q. So just to tell you briefly a bit more about the APC model, as I mentioned, this is a conditional model. We induce deletion by inducing expression of MX1. And the mice succumb rapidly to a macrocytic anemia and have some of the dysplastic features such as dyserythropoiesis and erythroid dysplasia. So our studies demonstrated that APC was essential for maintenance of stem cells and that haploinsufficiency impairs hematopoiesis. So we also asked the question, what happens if you add additional mutations, such as a second gene on 5q, so you have haploinsufficiency for two 5q genes, or add P53? And what we see is that the addition of additional 5q genes accelerates the development of MDS. And we also, I won't show you the data, but we also showed that ENU treatment accelerates the development of disease. So thinking about the role of APC then prompted us to look a bit more into the emerging role of Winch signaling in hematopoiesis. And it turns out that there's very good evidence that Winch signaling plays a role in the bone marrow microenvironment. It is expressed in mesenchymal stromal cells and in osteoblasts, and it regulates the differentiation and function of these cells. If you overexpress beta-catenin constitutively, it leads to AML in mice, and Winch signaling is activated in mesenchymal stromal cells and osteoblasts in some MDS and AML patients. But what is the role of Winch signaling in hematopoietic cells? Well, it turns out that Winch signaling in the stem cells themselves is essential for regulating self-renewal and quiescence. And stem cells are exquisitely sensitive to the levels of signaling. If you have increased signaling, you have bone marrow failure, and if you have decreased signaling, you have bone marrow failure. And it's activated in multiple subtypes of MDS and AML. So the next question that we asked, with our APC model, one of the first experiments we did once the mice developed MDS was to see if the disease was transplantable by transplanting hematopoietic cells from diseased mice into healthy mice. And we found that it was not transplantable. So that immediately raised the question to us, is this caused by a microenvironment effect as opposed to a cell-intrinsic stem cell effect? So to ask that question, we took bone marrow cells from a variety of different mice, including APC deficient, EGR1 haploinsufficient, and P53, as well as wild type cells, and transplanted them into recipient mice that had an APC deletion or wild type. And what we found was that, that only when we put, no matter what type of cells, whether it was wild type cells or APC deficient shown in red, when we put them into an APC deleted recipient, that was the only time that we got disease. And when we put disease cells into a wild type recipient, the mice did not develop myelodysplastic syndrome. And similar results were obtained with all of the genotypes that we transplanted into these mice. So that then suggested that, that it is a diseased microenvironment that's influencing this disease. So then we asked the question, what is the status of APC in the bone marrow niche cells themselves? So we isolated mesenchymal stromal cells from the bone marrow and showed first that yes, that the floxed allele of APC is actually deleted in mesenchymal stromal cells. So I think the point is that we make these nice models with things like MX1 Cre, and we assume that they're conditional and tissue specific, but that there are multiple other cell types in the body that are actually being affected and deleted, maybe subject to the genetic deletion. And then looking at the level of beta-catenin, we confirmed that haploinsufficiency for APC does indeed lead to a reduction in beta-catenin in these cells. So this was a little surprising to us because, of course, we assumed that everything was happening in the hematopoietic stem cells. So the next question we asked is, APC has multiple roles. As I mentioned earlier, it plays a role in cell migration, in metastasis, in chromosome segregation. So was it APC's role in Wynt signaling that was critical for this disease? And to ask that question, well, excuse me, I'm getting one step ahead of myself. We first asked the question, there's very intricate crosstalk between bone marrow cells, between hematopoietic stem cells and the cells in the niche. And we wanted to ask the question, does aberrant Wynt signaling in the niche alter these interactions? And is it genotype specific? So we did that by transplanting various genotype cells into either APC deleted mice or wild type mice. And regardless of the genotype of the cells that we were using in combinations of mutations, we saw the exact same effect. So the effect does seem to be bone marrow specific. It does seem to alter interactions with all types of hematopoietic cells of the various genotypes relevant to deletions of 5q. So the conclusions from this part of the talk is that APC-induced MDS is extrinsic to the stem cells, and it implicates Wynt signaling in the niche in myeloid disorders. And we also, in studies I haven't had a chance to present today, we also showed that cytotoxic therapy accelerates the onset of myeloid disease. And we believe it's affecting both the niche and the stem cells, and I'll come back to that point later. So now we ask the question, is APC loss in the niche mediated by the Wynt pathway itself or one of the other functions of APC? And to ask this question, we created mice that were doubly heterozygous for APC and beta-catenin. And what we found was that loss of beta-catenin completely rescued the development of MDS in these mice. So that did suggest then that, that the effect was being mediated by signaling through beta-catenin and through the Wynt pathway. And this just illustrates some of the hematopoietic feature of these mice, showing that the doubly heterozygous APC and beta-catenin mice shown in blue have normalized red cell counts and normalized MCV, RDW, and hemoglobin. So we asked the same question by doing a transplantation model so that we could specifically look at the effect in the bone marrow, in the niche cells themselves, as opposed to the hematopoietic stem cells. And what we found is that the mice who had a genotype in the bone marrow niche that was the double heterozygote for APC and beta-catenin haploinsufficiency, shown in blue, had much delayed development of disease, but we did see MDS developing in about half of those cases. So we asked the question a second way, and that is, could pharmacologic inhibition of Wynt activity abrogate the phenotype? And to do that, just to remind you, in the absence of Wynt signaling, the APC destruction complex proteins, such as casein kinase 1A, which begins the process of phosphorylating beta-catenin, which ultimately is then recognized by an E3 ubiquitin ligase subunit and targeted for degradation. When you have, in the presence of Wynt signaling, phosphorylation of beta-catenin is inhibited, and so beta-catenin is stabilized. And to look at this, we used a drug called previnium tosylate. And this binds to a casein kinase 1A and actually activates it, leading to beta-catenin degradation and inhibition of the Wynt pathway. Previnium is a drug that is FDA approved for treatment of pinworms and has very few side effects. So in the presence of previnium, which binds to casein kinase 1A1 and activates it, as I said, the phosphorylation of beta-catenin is stimulated and it's degraded. So to do this, we took cohorts of mice that were Cre negative, so essentially wild type, and mice that were Cre positive, so had heterozygous for APC, treated them with either DMSO or previnium tosylate. And the mice who were treated with DMSO, shown here, died rapidly as expected of MDS. And at the point when all of these mice had died, the mice that were treated with previnium survived and had no hematologic defects. We stopped treating we stopped the treatment, and those mice progressively then developed MDS, and that is shown here by their hemoglobin levels. You can see they slowly and progressively develop disease. So this suggests that a pharmacologic inhibitor of Wynt signaling would prevent the disease. We asked whether there was a dosage effect, so whether this was sensitive to Wynt signaling levels in the microenvironment. And so we did the same experiment using transplantation of wild-type cells into APC niche-deleted mice and treated them with DMSO, or a very low dose, 0.01 milligrams of previnium, and saw that these mice succumb rapidly, but then at higher doses, we could see a prolonged survival. And what I haven't shown you is that we were actually able to show that by isolating then the mesenchymal stromal cells from these mice, we asked, are we hitting the target? Are we hitting beta-catenin in these? And you can see that in PT treatment of the mesenchymal stromal cells decreased Wynt-mediated elevation of beta-catenin in both cytoplasmic and nuclear levels of beta-catenin. It also suppresses the expression of Wynt target genes, LEF1 and AXIN2, but not a control gene. So then we asked the question, is previnium useful in treating mice who have already developed anemia? And that is shown here, where we took mice that had anemia and treated them with DMSO or previnium. And the mice treated with DMSO developed MDS rapidly as expected, but the mice who were treated with previnium had a significantly prolonged survival. So we then asked, if you look at the hemoglobin levels just as a measure of the severity of anemia in these patients, in these mice, is there a difference between the survivals? And we found that mice who had a more severe anemia had a shorter survival, so there was a significant relationship with the level of anemia. So this suggests that previnium may be effective, particularly in the earlier stages of MDS in prolonging the development, delaying the development of this disease. So how does this relate to patients who have myeloid disorders with deletions of chromosome 5 or other myeloid disorders without deletions. And what I'm showing you here is just a panel of mesenchymal stromal cells that we isolated from our patients with or without deletions. And this just shows you 2 patients who did have deletions of chromosome 5. And I want to point out that in the presence of Provinium, that we do see a decrease in beta-catenin levels, both cytoplasmic and nuclear, and we see a decrease in the expression of, in this case, axin, CCND2, and TCF1, but not the control gene. So we and others have shown that there is a activation of beta-catenin in a subset of MDS and AML patients in the osteoblasts and mesenchymal stromal cells in the bone marrow niche. So the conclusions from this portion of the talk are shown here. Our work has shown that Wnt signaling in the bone marrow niche is critical in hematopoiesis, and that elevated signaling results in ineffective erythropoiesis and the rapid onset of MDS. We've also shown that APC functions in the bone marrow niche through the Wnt signaling pathways, and that inhibition of Wnt, either using genetic models or pharmacological inhibition, rescues the MDS phenotype and the development of anemia. And this raises the question of whether or not targeting the Wnt pathway in combination, most likely in combination with other therapeutic approaches, might be an effective approach in myeloid disorders. So I'd also like to turn now to talk about the role of Wnt signaling in the hematopoietic cells themselves. And an elegant work done by others, Luis et al. Showed that Wnt signaling is hematopoietic cells are exquisitely sensitive to the level of Wnt signaling, in that when you have increased expression above normal, you have impaired hematopoietic stem cell self-renewal and differentiation. If Wnt signaling is too low, you have enhanced hematopoietic cell function. So we asked the question then, what is the status of Wnt signaling in therapy-related myeloid neoplasms? And of course, we initially focused on looking at patients who had deletions of chromosome 5 because we were predicting that those patients who had loss of APC would have aberrant Wnt signaling. So the first thing we did was to look at the distribution of genes that are involved in positively and negatively regulating Wnt. And we were really surprised to find that there is an overabundance of genes on chromosome 5 that regulate Wnt signaling. And it turns out that the negative regulators of Wnt signaling are shown in green, and you can see that there are quite a few of genes encoding products that negatively regulate Wnt. And a few are positive regulators of Wnt, shown in red, and then a few others are shown in blue, are Wnt responsive genes. And that's just illustrated here. The genes shown in green are the genes that are negative regulators of Wnt signaling. So one would propose then that in a haploinsufficient state, with decreased expression of those genes, that you would have increased Wnt signaling. So we looked at Wnt signaling in our patients who have therapy-related, I apologize for the change in the fonts here. This is the group of patients who have deletions of chromosome 5, and I'm sorry, we're missing a panel there, but what we found was that the negative regulators are down in patients with deletions of chromosome 5, so that's consistent with our hypothesis that loss of 5q results in loss of expression of the negative regulators, and the positive regulators of Wnt signaling are up. So that would suggest that there is activation of Wnt signaling in these therapy-related myeloid leukemias. So we next sought to develop a mouse model for deletions of chromosome 5 by looking at combinations of genes. So taking what we believed were some of the most critical genes on 5q, giving rise to the phenotype, and looking at haploinsufficiency along with, in this case, P53, which is characteristic of del 5q leukemias. And we did this using a transplant model where we took haploinsufficient cells for EGR1 and APC and infected them with short hairpins for P53 and then transplanted them. And what we found was that a very small number of these mice developed myeloid neoplasms. And that's just shown here, the appearance of the myeloid disease. But interestingly, these recapitulated some of the features of del(5q) AMLs, having a very complex karyotype and an aberrant double-strand break response. So thinking back to the patients, we thought about what is different in our mouse models and the patients. And of course, we have to recognize that patients are treated with cytotoxic therapy, and that in the human setting, both the niche as well as the hematopoietic cells are exposed to that therapy. And what are some of the possibilities? The first is that the cytotoxic treatment induces mutations in the hematopoietic stem cells. Another possibility is that there are pre-existing mutations in stem cells, the CHIP phenomenon that we talked about, and that cytotoxic therapy results in clonal selection, and that permits the acquisition of secondary mutations in leukemogenesis. Another possibility is that the bone marrow niche is influenced by the cytotoxic therapy, and that this creates a permissive stromal cell niche that enables the expansion and survival of these rare mutant stem cell clones. This could be the effect of epigenetic alterations, an inflammatory response with cytokine secretion, changes in oxidative stress, kind of a more global phenomenon in the bone marrow. And you recognize that these are not mutually exclusive, and that both of these could be happening. So let's just focus for a moment on the concept of clonal selection in the hematopoietic stem cells. The idea here is that somatic mutations occur in stem cells, increasing with age, and that some of these mutations confer a fitness advantage. There are competitive conditions that would then select for these mutated clones, things like immune destruction or dysfunctional hematopoiesis in MDS, chemotherapy or radiotherapy, toxins or drugs, inflammation, infections, may select for and create a clonal advantage with loss of the normal hematopoietic cells and survival of the mutated cells. Those cells may acquire additional mutations, secondary changes, and go on to develop into malignant diseases. But an interesting point with this model is, if you think about it, these clones have already undergone selection. They've already undergone exposure to cytotoxic therapy and undergone selective process, so they may have inherent resistance to therapy through this process. So we asked the question then, in our mouse models, what would happen if we treated these transplanted mice now with ENU, and we treated both the donor and the recipient. And what we found is that 82% of these mice now developed AML and MDS with multilineage dysplasia. That's characteristically very similar to human therapy-related myeloid neoplasms. So then we asked the question, is P53 essential for the development of AML? And so we used a control short hairpin instead of P53, but again treated both host and recipient with ENU. And what we found is, yes, P53, in this case, the mice all developed MDS with dyserythropoiesis, but none of them developed AML. So P53 is required for the development of AML. So in this first experiment, the first experiment I showed you, where only 20% of mice developed AML, the mice had not been treated with ENU. When we treated both the donor cells and the recipient with ENU, 82% of the mice developed AML. But this does not allow us to separate the role of the ENU treatment of the hematopoietic cells or of the niche. So we did a separate experiment where we treated only the recipient, so the niche cells were exposed to ENU, or the reverse, where only the hematopoietic donor cells were treated with ENU, but not the recipient. And we predicted from this analysis, based on the CHIP phenomenon, that we would see an increase in expansion of the P53 population. But what we found was that there's a very small expansion when mice are not treated with ENU. when you treat the recipient or the donor, you get a slightly increased expansion of P53 cells. But it's only when you treat both the donors and the recipient that the niche and the hematopoietic cells that you see a significant expansion of P53 deficient cells. And it's only when we treat both the niche and the donor cells that we see the development of AML. When you treat only the recipient, shown in yellowy tan, or the donor, we only saw AML developing in one mouse. So this suggests that there are synergistic effects, there are effects of cytotoxic therapy on both the bone marrow microenvironment and the hematopoietic cells, and that there may be synergistic crosstalk between the two that give rise to the development of this disease. So just to finish up, we have done sequencing analysis of the myeloid leukemias arising in these mice. And what we find is, interestingly, there appear to be similar number of mutations in mice that have either ENU treatment or no ENU, or P53 or no P53. But the important difference is that in the mice that are treated with ENU, what we see is an increase. If you look at the red boxes, in these mice, we see a significant increase in mutations in genes that are known to be involved in leukemogenesis. So although we see mutations in the mice not treated with ENU, they tend not to be in known leukemogenic genes. But importantly, we also see that there's a pattern where we see mutations in genes that are involved in the DNA damage response or DNA repair in mice that have been treated with ENU. And we see that is also shown with expression analysis, where we see increased DNA repair pathways, and this just shows one DNA repair pathway showing you that there's an increased expression of genes involved in that pathway in homologous recombination. So this has also more recently been reported now in patients with therapy-related AML, where we don't see mutations per se, but we see an increase in overexpression of genes that are involved in DNA repair. So just to summarize our models, what we've shown by looking at combinations of loss or haploinsufficiency for combinations of genes, 2 genes on 5q and P53, that it's only with the combination of all 3 genes that we see that we can recapitulate the phenotype of therapy-related AML with trilineage dysplasia, increasing myeloblasts, the development of AML, and complex karyotypes. So EGR1 and APC haploinsufficiency promote the development, but we see increased severity with loss of more than 1 gene on 5q and loss of P53. And P53 was critical for development of frank leukemia. And that we believe that development of TML is probably promoted by the effects of cytotoxic therapy on both the hematopoietic cells and the bone marrow niche. So just to summarize the pathways that lead to therapy-related AML, I just want to emphasize, I think that there's both intrinsic and extrinsic factors that contribute. There's inherent patient-related germline factors, but also external factors such as the nature of the disease, the cytotoxic treatment, comorbidities, environment, et cetera. But I think there's multiple pathways that we can think about. The first is that there's an inherited predisposition mutation such as BRCA1, and exposure to cytotoxic therapy, there's a defect in DNA repair leading to the acquisition of potentially of mutations and development of the disease. An alternative pathway is that exposure to genotoxic agents, in this case I'm showing the topo2 inhibitors, which are believed to be directly involved in resulting in DNA breaks that give rise to the recurring translocations that are seen in this subtype of therapy-related AML. The third model is clonal selection, the CHIP phenomenon, where you may have mutations such as P53, chemotherapy and radiotherapy-induced clonal selection, you have additional somatic alterations and the development of disease. And finally, the abnormal microenvironment, where you have defective stroma, and that may pre-exist, or it may be induced by chemotherapy and cytotoxic therapy, creating a permissive environment that leads to the acquisition of mutations in stem cells and the development of disease. And of course, you recognize that these are not mutually exclusive, in that several of these could occur at the same time. So just to finish with some of the unanswered questions, I think we don't really know yet the mechanism for clonal dominance or fully understand the characteristics of the leukemia-initiating cells. We don't know the full spectrum of underlying genetic susceptibility to therapy-related AML, or the role of CHIP. Which genes and mutations or combinations confer the highest risk? In what context? And can we use CHIP as a biomarker? And we're still in the process of defining the genetic pathways and cooperating mutations that lead to the disease and progression and drug resistance. But our work and that of others have led to therapeutic implications, and that is, can we identify high-risk patients? Can we reduce the risk of therapy-related AML? And one of the things that others are looking at is, can we target the niche? For example, by decreasing Winch signaling or inflammation, or P53 mediated stress response to create a healthier environment for hematopoiesis. Can we identify molecular targets, one being the Wynt pathway? Or can we target vulnerabilities due to haploinsufficiency, as Ben Ebert and his colleagues have shown using lenalidomide, that targets haploinsufficiency for casein kinase 1A1, a gene that's deleted on 5q. But one of the things we're excited about is that we think with our models we've been able to develop some tractable models for preclinical studies, and we're proceeding with studies looking at other Wynt inhibitors, but also combinations with conventional therapies that are currently being used for AML and MDS. And I will close here by just thanking all of the individuals, particularly Angela Stoddart in my laboratory, who has led this work. And I'm happy to close and answer a few questions. Yeah, so in our simplistic way of thinking, what we're doing first is to sequence stromal cells from the mouse model where the mice have been treated with the NU to try to get a sense of what might be happening in that model. We've also been isolating stromal cells from patients and plan to sequence those and look at those. That's a little hard. It's a little harder to grow those, and I also worry about what population of stromal cells we're growing. We're also, so in the back of my mind, I think that some of the same things that are happening in therapy-related AML may be happening naturally as we age, and so the AML and MDS that we see in the elderly may also be related to changes in the bone marrow niche. So some of the other things that we're doing are trying to grow stromal cells that we get from hip replacements and other samples that we can grow from younger and older individuals, and try and characterize differences in the stromal cells and bone marrow cells in those patients. So that's a long way of saying I don't exactly know what it is doing. I think it can be doing a variety of things. I don't know if it's inducing mutations. I kind of suspect not. I actually think it may be in the stromal cells, and it might be in inducing more of a global phenomenon such as epigenetic changes or inflammation, inflammatory responses, reactive oxygen species. So something that's a more global physiological consequence. Yeah, that's a great question. So I would say that in myeloid diseases, what we've learned is that the deletions of chromosome 7, deletions of 20, also involve haploinsufficiency. So this may be a more common effect in myeloid disorders. I would raise the possibility that these large deletions are also characteristic of solid tumors, and we haven't really touched on that phenomenon in solid tumors, but it may also be these contiguous gene syndromes may be occurring in solid tumors. To get back to your real question, there's clearly a selection process going on. If you have a big deletion of another chromosome that doesn't have any critical genes, you're never going to see the leukemias arising. I believe that recombination occurs at certain sequences, that there are certain regions of the genome that are more prone to recombination events. It's not a 100% perfect model, because if you look at SNP arrays, others, Masiewski at Case, have done hundreds of patients with deletions of 5. And they can show that the breakpoints, there are some common breakpoints, but it's not 100%. We don't see the same breakpoints in 100% of patients. So that model could still be true. Chromosome 5 does have a whole cluster in Q13 where in Q14 where we see a lot of the breakpoints has a huge cluster of the mid-level repeat sequences that are specific to chromosome 5. You may be aware that there are some mid-level repeats that are unique to different chromosomes in our genome, and there is a cluster there. So it may be that we're getting recombination events. I've always suspected that the alkylating agents, cytotoxic therapy, maybe radiation therapy, is causing double-strand breaks that then results in religation and recombination events to give rise to the deletions. Oh, that's a great question, and I have no idea. I mean, the genes that we're seeing cover a huge region. They cover about 70 megabases. So I have trouble understanding how that would work, but I think it's more a concurrence or an unfortunate event that you have a whole cluster of genes that are critical in hematopoiesis that happen to be located in the same region. Right, so I will answer that 2 ways. Yes, some of the studies looking at the AMLs, more recently in Chernobyl, the AMLs that arose in those cases did have similar abnormalities, a high risk loss of 5 and loss of 7. So that would suggest a link. The second way I'll answer your question, I know you're not going to like me saying this, but we have about 90 patients now in our series of therapy-related AML who received only radiation therapy and no other cytotoxic treatment. And a high proportion of prostate cancer patients. And when we look at those cases, they look almost identical to the high-risk therapy-related group. They have abnormalities of 5 and 7. They have clinical high-risk features. Is that because of the therapy, or is it because it's an aging population and they're developing a high-risk AML in the elderly, typical of the elderly anyways? I can't answer that. I can only say that we have a pretty high proportion of patients in our series.
Okay, great. Thank you.
Well, thank you.